Hi, we're Team Restor3D and we're here to talk about our Capstone project, the printed photopolymer cranioplasty implant. We are 5 graduating seniors consisting of 3 mechanical and 2 biomedical engineers. We have had the pleasure of working with Dr. Nathan Evans and Ben Wesrick from Restor3D. Cranioplasties are a surgical procedure that involves reconstructing the human skull by fixating a cranial implant on a patient to both shield the brain and reconstruct proper skull contour. Working with Restor3D and 3 surgeons, we identified a need for a patient-specific cranial implant that addresses expensive material use, excess waste production, surgeon user needs, and no FDA 510(k) clearance. Our challenge is to develop a patient-specific cranial implant design and manufacturing workflow capable of producing an FDA-compliant device that satisfies customer requirements.
In conducting a prior art search, the team first researched cranial implants currently on the market. Additionally, a patent search was conducted to identify the key aspects of intellectual property being claimed in this space. Cranial implants on the market can cost as high as $12,000, and as the market continues to expand, it's overdue for a competitive innovation. We have created a novel design and manufacturing workflow for production of the first FDA-compliant cranial implants made from photopolymers.
The implant design process starts with converting the patient's CT skull scan into a 3D model. The skull is then mirrored and an implant is created to match the missing bone. Once the implant model is completed, it is set up in the 3D printing software with proper supports and printed with stereolithography technology. Once printed, the implant is bathed in an alcohol bath, removed from the build platform, and cured with ultraviolet light and heat to strengthen and complete the chemical processing. Finally, the supports are removed, extraneous material is filed away, and the implant is ready to be sterilized, packaged, and sent for implantation. We have simulated a surgical procedure with a 3D printed skull and brain phantom model. First, plates are screwed into the implant's printed screw holes on the side table. Next, the plates are screwed into the patient's skull with safely sized screws. Finally, skin is pulled back over the skull. The absence of such a large section of bone is easily visible through the skin. The addition of the implant protects the exposed brain and provides symmetry back to the skull. The design inputs are pulled from both surgeon requirements and FDA requirements to prove substantial equivalency to a predicate device. The most consistently requested features of the implant were modifiability during surgery, a quick turnaround time from scans being sent to the implant being returned, and utilization of standardized fixation with plates and screws. The predicate device chosen then dictates the required mechanical and biological properties of the material and design needed to obtain FDA clearance. The FDA 510(k) regulatory strategy begins with identifying a suitable predicate device. We have chosen MedPore, a porous, high-density polyethylene material from Porex. Then testing is performed on the predicate material and our proposed material. The MedPore 510(k) submission included tensile, bending, and biocompatibility testing. Due to the length and cost of biocompatibility testing, we decided to focus on the mechanical testing first. Finally, a justification must be made using testing data and engineering analysis that the proposed material and the design is substantially equivalent to the predicate.
We spoke with 3 surgeons during this project, Dr. Gonzalez, Dr. J. Kumar, and Dr. Lee. From this, we learned that they use plain screw method of fixation, but they're also interested in tabs. They're typically provided with 2 implants, one for planning and one for the actual procedure. They prefer an implant that has a thickness of less than 5 millimeters, that the screws they use are 1 to 2 millimeters in outer diameter and 3 to 5 millimeters in length, and that the material needs to be modifiable with surgical tools. One part of our testing process is testing the simulated use of our device based off the requirements we defined. We tested screwing into plain material, testing how hole sizes changed when the implant was printed, if printed tabs are feasible, how to screw into holes when they're on the implant, and how much torque it takes to screw into various hole sizes. This testing had a mix of qualitative and quantitative results, and it was an important step in guiding our design choices.
In our case, the predicate performed tensile and bending testing, so these were the ASTM standards used for testing. The tensile testing results show that the nominal ultimate tensile strength value of Biomed Clear is about 50 megapascals larger than that of the MedPore predicate. The bending testing results show that the nominal flexural strength value of Biomed Clear is almost 100 megapascals larger than that of the Medport predicate. The novelty of the implant is that it is the first FDA-compliant cranial implant design made from photopolymers. It offers a 21.4% reduction in cost even when factoring a 10-time markup that is often used in the medical field, and the usage of additive manufacturing allows for a 77.5% material waste reduction when compared to other implants on the market. As far as next steps go, we'd like to explore new fixation mechanisms to simplify implantation even more. Additionally, we would like to coordinate with Restor3D to continue progression of the project and submit 510(k) for FDA clearance. Finally, we'd like to acknowledge and offer many thanks to those who have been involved throughout our project.